Okay, so we just ended on factors which don't affect enzyme activity. So temperature does, it denatures our enzymes. Prosthetic groups, you add those in. Phosphorylation does something as well, but glycolization, that's for post-translational modification. Cool. I think this is the last question. Enzymes increase reaction rates by what? This is also channeling 3-4 Biochem.
Anyone take a shot?
Get some chat. Hey, good stuff. So we decrease the energy of activation by providing an alternative energy pathway. That's regurgitated from my brain somewhere. But pretty much an enzyme will hold a molecule in place so that reaction is able to take place. Inhibiting the rate of reverse reactions, nah. It's if an enzyme would catalyze a forward reaction and a reverse reaction, they don't really impact on the reverse when we're going forward. Changing the equilibrium constant, oh, bruh. So I think we talk about equilibrium constants maybe in terms of like pH and some Chris Wright stuff or something.
Yeah.
Maybe pharmacokinetics. Anyway, it's not in relation to enzymes, so don't worry about that. And altering the change in free energy of the reaction. So you can't change free energy because any energy that's created can't be destroyed, essentially. Either lost as heat or it's translated. Cool. I think that's it for this one. So I think
Any
next we've got
questions?
fuel molecules.
Any questions before we move on to fuel?
Yeah. Has this session been have you been following all right then? Just get like a yes in a yes reaction or something like that. Yes, no, yeah.
Love hearts to you in chat as well.
Yeah.
Awesome.
All right, cool.
Awesome.
Hi. So
Alright, so
to share my screen, can
That's us.
somebody?
Oh, Nikita, do you want me to share? Oh.
It says I've already got it up, but I'm just unable to because I'm an attendee and not a host.
That's okay. I'm making you the host right now. Okay. You should have access right now.
Yeah. Google Chrome. Okay. Is that all okay for everybody?
Yep, that seems to be working fine. It is kind of splitting the screen slightly, but should be fine. Yep, it's working all right.
it's meant to me.
I was just testing the other day. Don't worry about me.
Thank you for the smiling face.
Yeah.
Okay,
Perfect.
so metabolism is quite a high yield unit compared to some other stuff. Majority of the questions on the exam are biochemistry related, so it's quite important. Is just what I'm trying to.
Yeah,
Okay.
I'll be presenting these slides too. So.
Yeah.
But Nikita made most of these slides, so she'll probably present most of it, but I'll chip in here and there. Okay, so this is kind of just a big flow chart that I made last year, basically covering a lot of metabolism. Please try not to panic about this too much at the moment. You will have to know all of it eventually. Yeah, it's basically like a big overview of what we're going to be going into. I know it's really big and you can't like go back to back. Nikita, come back.
Why do we need metabolism?
All right.
Essentially, the whole goal of
All right.
life is to survive and reproduce. So you need ATP to carry out your daily tasks. Pretty simple.
Nikita.
Yes.
Could you go back to? Yeah, go back one. Yeah. So basically, when you're learning metabolism, I want you to keep in mind that like all of these steps here, right? If I can draw, like all, yeah, okay, like all these steps here, blah blah blah, right? So the main goal of all of these steps, right, is to produce ATP, right, which is over here, right? So basically, these steps don't, they're not like that. They're important, right? But then they all lead to this step, right? This step is the main important part, right? Because this step is like the step that produces the most ATP. So that's the step that you want to get to, right? So that's just something to keep in mind. So the main goal of all these other like pre-electron transport chain steps is to get to this step, this final step right here.
Yeah,
That's the one-
essentially to
one I
get
want
to
to
the ETC.
Yeah,
Cool. Let me know when you want me to change the slide.
yeah, go.
So before we get started, I just wanted to break down everything in terms of what you need to know. So breakdown is catabolism. That is breaking down big molecules into smaller molecules. That is using water. So that's hydrolysis. It's releasing energy, and I like to think of cellular respiration. And then in terms of buildup, that's anabolism. So smaller molecules to bigger molecules. You're releasing water. It's using energy, so it's endergonic, and then photosynthesis. So that's light energy coming in, and then you're watering plants, and they are also taking in carbon dioxide to produce the bigger molecule, which is glucose and water. Is that all good?
Yeah. If you guys ever heard of like anabolic steroids, so like, you know, like if you want your muscles to get like really big. Okay, I don't know what I do. But basically, yeah, those are what anabolic steroids do. Like,
They build up your muscles.
yeah, build up pretty much smaller molecules to bigger molecules. So that's what I like to think about, just to help me remember.
So essentially, how do we get glucose? So you eat it, right? You eat your carbohydrates. Or what happens is that usually when you have too much glucose in your bloodstream, there's a signaling molecule, and that causes glucose to be converted into glycogen, and you store the glycogen. So in terms of getting glucose already from your body, you're converting the stored glycogen into glucose. So this is a cellular respiration flowchart. Most of my knowledge was based off biology 3-4 on this. So essentially the way I remember it is with glycolysis, you have the conversion of glucose, a 6-carbon molecule, into 2-pyruvate. So that's 2 3-carbon molecules. And this usually occurs with or without oxygen. So we're not talking about an oxygen limited or an oxygen excess environment. You can see the inputs and outputs there, but it does produce, it happens in the cytosol of a cell. So if we go and look at glucose, essentially it's being converted into pyruvate here. What the most important thing to recognize here is in the absence of oxygen, we get pyruvate formation. So that's converting NAD plus to NADH. But what happens is essentially if we have no oxygen, we produce lactate. And this NADH is then converted into NAD plus. And this means that we can keep on producing pyruvate and hence keep on producing the ATP that we want. Does that make sense? Or would Ben like to add anything?
Would you like to explain why NADH is important?
Would you like to do that?
Yeah, sure. So, yeah, so go back one slide.
Yes. Actually, how do I go back? I think
back. Okay. Okay, maybe go back to the second slide,
Yeah.
like the big flow chart.
Hang on. Sorry, this is being really clumpy.
See, this is why you should have given the share screen to me. Alright, whatever. Yeah. Anyway, okay, so NADH is a really important molecule, right? I mean, sorry, not NADH, NAD plus, right? Wait, am I? Yeah, NAD plus, right? And the reason for that is because if you look at this step here, right? So I don't know if you can read it, but this step of the citric acid cycle, right? In order to produce NADH, you need NAD plus, right? So that means that if you don't have any NAD plus, then that means you can't go through this step, right? And if you can't go through this step, then that means you can't go through this step, and then you can't go through this step. And that means you can't produce ATP, right? So NAD plus, right? So that's a very important molecule for you to have, right? But sometimes you might have too much NADH and not enough NAD plus, right? So that means you can't go through this step right here, right? And what this over here does, what anaerobic respiration does, is that it allows you to convert some of your excess NADH into NAD plus, right? So like this step, like it doesn't produce any like ATP by itself or anything, but basically what it allows you to do is it allows you to go through this step. So allows you to produce some NAD plus to basically let you go on with respiration, producing ATP. Yeah, that's all I wanted to say.
Okay, yeah. So that was anaerobic
Jump
respiration.
in as well.
Mm-hmm.
Sorry, just with NADH, it's a cofactor, so we use it in relation to enzymes, and it can be loaded and unloaded. So that's that cycle that Ben was talking about, where it's either NAD plus, adds an H, then it's NADH, and it goes back and forth in a circle. Cool.
Okay, so then we have anaerobic respiration. So that was what I was explaining before, where glucose is converted into pyruvate and then lactic acid plus 2 ATP. Obviously, this is reversible in the presence of O2 because lactic acid is an aqueous solution, so there's nothing which is really escaping. After that, what we have, so there's a question, is NAD plus the cofactor or NADH? Could you answer that, Will?
I think he's muted.
Big brain, I was muted. Anyway, so the NAD plus is the cofactor, and then it's loaded with an H to become NADH. So the NAD plus itself is the cofactor. Cheers for that.
In terms of glycolysis, what you need to know is what I have here. So don't memorize it, but there's 10 steps in glycolysis. So essentially steps 1 and 3 require 1 ATP each. Steps 7 and 10 also produce 1 ATP each for each 3 carbon. So that's essentially they're producing 2 ATP each. So then steps 1 and 3, 7 and 10 combined produce 4 ATP, and the net total is 2 ATP. Then you have step 6, which produces 1 NADH and a hydrogen ion for each 3 carbon molecule. So you don't need to know that in a lot of detail, but it's just good to be aware of it. And then further on, what happens is glucose plus ATP produces glucose 6-phosphate plus ADP. So what that's happening there is that it's essentially important because glucose 6-phosphate comes up in muscle cells. So it's an important one to be aware of in terms of what it is. And then another thing you should know is that step 4 is essentially splitting up a 6-carbon molecule into 2 3-carbon sugars. Yeah? Any questions? Cool. The next one is the Krebs cycle. So what we have in the Krebs cycle is you have pyruvate and you've got 2 pyruvates. So this cycle, I'm pretty sure it happens twice, once for each pyruvate, or twice for each glucose molecule and once for each pyruvate molecule. So there's a transition step before the Krebs cycle, and what's happening there is that pyruvate is being converted into an acetyl-CoA and a carbon dioxide. So obviously carbon dioxide has 1 carbon, but you're producing 2 of them. So I think that's counting in the 2 plus the acetyl-CoA. And that adds up to 3 in terms of the number of carbons. What happens then is you have 2 acetyl-CoAs, each of them enter the cycle, and you produce 6 NADH in total and 2 FADH2. So first in glycolysis, we produced our NADH, and now here we're producing our NADH and FADH2 molecules, and all of these are going to end up going into the ETC to produce the energy that we need. This happens in the mitochondrial matrix. I already said it happens twice for each glucose molecule. The enzyme which converts pyruvate to acetyl-CoA is called pyruvate dehydrogenase. And an important point here, which is an addition upon Bio 3.4, is that enzyme requires thiamine, which is a B group vitamin. And I'm pretty sure that is acting as a cofactor for that reaction. If that all makes sense. Anything to add, Ben?
No, not really. Yeah, you can keep going. I don't have anything.
The next part is ETC, and it's important to remember with the Krebs and ETC, it's not only glucose which can enter them, but there's also a way of getting fat to be converted into acetyl-CoA and then contribute to producing energy as well, which is something that we will be looking at later. So in terms of what's happening here is that you're putting in oxygen, you're getting out water, and you're getting out a lot of ATP. There's 4 different complexes in the mitochondrial cristae or the inner membrane. So at complex 1, I'm just going to try and annotate here if I can. So complex 1 is over here, so I just circled that one. What's happening is the NADH and the FADH2, which entered from your glycolysis and Krebs, is what's reacting here. So first complex, you've got NADH being converted into NAD plus a hydrogen ion. And then at complex 2, you have FADH2 being converted into FAD plus H plus, or 2 hydrogens, not that important. And at complex 3, what you happen is that the electrons which are being released at complex 1 and complex 2, so i.e. an oxidation reaction in terms of chemistry 3-4 content, you should know. It travels through complex 3, and at complex 4, what happens is O2 picks up those electrons, reduction, and it's converted into water. So the way that I really memorized this was that I used stuff from Chem 3-4 that I already knew in terms of oxidation and reduction to link everything together. The 4 large complexes tend to be electron carriers or integral proteins. And what happens after this is essentially that a hydrogen ion gradient is formed. So on this side, you have a high hydrogen ion concentration. So that's the intermembrane space. On the matrix side, you have a low hydrogen ion concentration. And from a rule of chemistry that we know is that everything moves from an area of high concentration, high solute concentration, to an area of low solute concentration. So hydrogen ions are pumped through this ATP synthase pump, exactly what it suggests. So synthesizing ATP. And then here what's happening is that ADP plus pi is being converted into ATP here. There might be something that I've missed. So would Ben like to explain anything?
No, not really. It's just that, like, I reckon this and this are the most important. You don't really need to worry about that. Just remember that NADH plus FADH2 is pretty much going through the series of steps here to produce ATP. That's basically the main takeaway you need to get from it. Yeah, he explained it pretty well.
This was a summary that Ben put up. I'm not to explain anything.
Did I put that up? I thought you put that up. Okay.
No, I didn't.
Whatever.
Okay, I'll just skip that. So yeah. Ben.
Oh, this is my slides. Okay. Yeah. So
I'll just give you host, Ben.
Sorry.
I'll just give you host, Ben, if you want.
Oh yeah. All right.
You don't have to.
Does that mean I'm screen sharing now? Oh.
Yeah. Yep.
No, Nikita's still screen
Oh, no, she
sharing,
can.
but
Yeah.
No, it's fine. I kind of see. We'll just stay in the key screen.
Okay,
anyway, so this is kind of just a bit of applied metabolism, right? So if we have excess glucose, we kind of want to kind of get rid of it to make some energy, right? So one way we can do that is through glycolysis, which is like this step here. Right. So this is the step of converting glucose into pyruvate, right? And I guess your body would probably want to go to this step when it's like low on energy, because if you can convert stuff to pyruvate, then you can convert stuff to acetyl-CoA, then citric acid cycle, electron transport chain. On the other hand, if your body if it already has enough ATP, right, and doesn't really want to make any more energy and stuff. It can also go through glycogenesis, right? So your glucose can be converted to glycogen, and that glycogen can basically be stored for when you need energy later on, right? So those are basically the two main steps that glucose can go through when you've like eaten like lollies or something or whatever, right? So if you've got enough energy, then you can go through this step. Otherwise, if you need some energy, you can go through this step. Next slide. Yeah. So if we have too little glucose, right? So like, so talking about like homeostasis and all that, right? So if our body detects that we have too little like blood glucose in our bloodstream, right? So the first step is that we can hydrolyze some carbs, like so like if we eat some bread or something, right? So we can our digestive system can break that down into glucose for us, and that's going to increase our blood sugar. The other step is that sorry, I can't see all this. Right. The other step is that we can also convert some of our stored glycogen into glucose, right? So remember how we talked about last slide, how if we had too much glucose, we can store it for glycogen for later on, right? So now if we have too little glucose, we can convert some of that stored glycogen back into glucose and get some energy from that. And this last step, you probably don't want to do because you kind of want pyruvate to go ahead with the electron transport chain. But I guess if you're running like really low in glucose and your body kind of needs to up your blood sugar levels, then this gluconeogenesis is kind of the step that we'll try to go through. And obviously it's not ideal because, yeah, as I said, you kind of want your pyruvate to go the other way. So ideally you'd want like to eat stuff, like either eat stuff to get your glucose or like convert some of your stored glycogen to glucose, but as a last resort, you can probably convert some of your pyruvate to, yeah. Yes, William. Did you have
I've just got with the gluconeogenesis, I'm like, correct me if I'm wrong, but another thing that forces that reaction to occur is alcohol consumption. You want to, yeah, I think year ones you'd talk about this maybe next week, but pretty much if you have too much alcohol, because of the imbalance of NADH to NAD+, it forces everything to go backwards and you make glucose from pyruvate. You go to gluconeogenesis rather than the other way.
Yeah, that sounds right. We'll probably go through more detail next week. Thank you, Will. Keto bodies. Oh, okay, yeah. So, that slide title is probably wrong. It should probably be saying like what happens if we have too little energy. But basically, what ketone bodies are is that they're kind of like a last resort source of energy, right? And one of the important things to note is that a lot of these exam questions might ask, okay, there might be like, oh, which one of the following is true, right? And then like, there might be like ketone bodies are a really inefficient source of energy, right? And that would actually be wrong. Ketone bodies are actually really efficient at like giving you energy. But the reason that they're a last resort is because they're really toxic, right? So if you have too many ketone bodies, your body kind of just dies, right? So you don't really want to be producing ketone bodies, but if you don't have enough energy, your body's going to produce some ketone bodies, kind of like a last resort. And yeah, they're really energy efficient, but you don't really want to produce many of them. All right, next slide. Yeah, okay, this stuff I reckon is pretty important, right? So we'll go through insulin first, right? So insulin.
Fire 3-4 negative feedback loop content, essentially.
Yeah, okay. So insulin is basically a hormone, right? And basically it's released when your body has too much glucose in your bloodstream, right? So let's say you had like a really big meal or something, right? Basically what it does is that it kind of tells your body to uptake your glucose, right? So get rid of some glucose from your bloodstream and store it in your liver, right? Through glycogenesis, right? So convert some of your extra glucose into glycogen for later, right? And yeah, it prevents, and what that does is that means that your blood glucose levels can remain at a relatively normal level. And if you ever heard of diabetes, right? So what diabetes is like, basically your body like can't respond to insulin anymore, right? Or at least like type 2 diabetes, right? And then people with type 2 diabetes would have high blood sugar, right? Because if their body can't respond to insulin anymore, that means that their blood glucose levels are just going to remain high because like you can't be storing, their body just can't store it for later. Yeah, glucagon's basically the opposite of insulin, right? So when your blood glucose levels are running low, what glucagon can do is that they can signal to your liver to break down some glycogen, right? And breaking down some glycogen is going to give you glucose. So yeah, so those two kind of work in opposite fashions, but they work they work together to try to ensure that your blood glucose levels are at a good level, not too high, not too low. And yeah, next slide.
Yeah. So this is essentially the mobilization of stored glucose. So glucose can be stored as glycogen in two places, so either in liver or muscle cells. And as Ben was explaining before, when glucose levels are low, it is signaled via glucagon and adrenaline to convert glycogen into glucose. So that glycogen is converted into glucose 1-phosphate or glucose 6-phosphate. So then what happens is in the liver, glucose 6-phosphate is hydrolyzed to glucose via an enzyme, and it maintains those blood glucose levels in the bloodstream via the negative feedback loop. Whereas in the muscles, the glucose 6-phosphate is kind of able to directly enter glycolysis, so it doesn't need to be converted into glucose, and the muscle cell can just directly use it for energy. That's a summary that you guys can go over in your own time.
Wait, stand aside. Yeah. So, yeah, it's pretty important to remember that, like, if you eat a glucose, if you eat like sugar or something and you get a glucose molecule, it basically goes through each of these steps in turn, right? So it goes through this step, and then that pyruvate here will go through the other, and then that creates acetyl-CoA, and then that acetyl-CoA will be converted to NADH and FADH2, and then that in turn will be converted to ATP, right? So through those steps, you can get ATP from the glucose molecule. It's just that like you can't go straight from glucose to ATP like that. You need to go through like each of these prior steps first in order to get ATP from glucose. Yeah, that's all I wanted to say.
Yeah, I think the next section you would have covered is lipid metabolism. So
wait, did you guys do lipid metabolism?
It was on the lecture when I looked at it. It was on the first lecture.
Just a heads up, we
yeah,
do have about 9 minutes left, so if you can fit it in that would be great.
Mm-hmm. Yeah,
Easy.
so this is stuff you can get over
Just like
your
how
own
it
time.
moves from the outside of the body inside, instead of like metabolizing it. So.
Uh-huh. You can go over this part in your own time.
Yeah, it's not really important.
This wasn't covered.
Wait, was it? Oh,
It's covered later. It's covered in their next lecture.
Oh, okay.
Yeah.
We'll go to that.
They've said they have covered it.
Oh, have they? Oh, okay. Maybe they've gotten through a few more lectures this week. I was told to do few
We'll
modules.
just go through this anyway. So it's like even if they haven't covered it, it'll be like a pre-learning. So anyway, so the word exogenous basically means like source from outside, right? So exogenous pathway basically means the pathway that your body goes through in order to metabolize dietary fat, or like fat which is sourced from like outside your body, right? That you've eaten. So if we go through, let's go to the next slide. I think these slides might be a bit out of order. It's just a bit of an explanation on exogenous pathway.
Do you want me to skip to it, or?
No, you can explain.
So essentially, why are fats better than using glucose as an energy source? They're more energy dense and they supply energy for long periods of time. How do we get fat? So we essentially eat them, or we can convert the stored fat in adipocytes. So those are fat cells. They're usually stored as triglycerides, and then we can convert them into 2 fatty acids and a monoglyceride that can travel through the bloodstream and provide energy. Fats can also be converted into acetyl-CoA and can enter the Krebs cycle. So essentially, the first thing we focus on is lipid digestion. So what do? We eat lipids, and then they end up in the stomach. The gallbladder ends up releasing bile. Then the bile emulsifies the lipids into micelles, which are smaller lipid droplets. Essentially, it's increased surface area, so lipase can act on them better. And then what happens is the pancreatic lipase acts on micelles to break it down into free fatty acids and monoglycerides. The free fatty acids and monoglycerides enter enterocytes. So enterocytes are intestinal cells. What happens is in the intestinal cell here, so the enterocyte, free fatty acids and monoglycerides have entered, and in the endoplasmic reticulum, they join up again to become tags. Tags then enter the cytosol of the enterocyte. They're packaged up into chylomicrons, or little vesicles, and these vesicles enter lacteals, so they're basically capillaries.
Explain what a chylomicron is.
Yeah, I'm doing that later.
Okay.
And
I don't know either, so.
Villi of small intestines via exocytosis. So they're just exocytosed out as chylomicrons, and chylomicrons can then enter the circulatory system to transport fat around the body. This is just a little nice diagram which kind of summarizes the whole lipid digestion process. It's visual and I prefer this, so you can learn via doing that as well. So in terms of lipoproteins, they're involved in transporting lipids. The core is usually hydrophobic, and that's because the lipids are insoluble, as well as tags and cholesterol esters. The external core is hydrophilic. The main reason for this is because it needs to be transported in the blood, and the blood is also hydrophilic. So the first type of, we have 4 different lipoproteins. The first type is chylomicron, so that's straight after digestion. It's in your intestinal cell and you release it. And it's mainly fat because we haven't delivered any fat around the body yet, and it has some cholesterol. So basically what it's doing is transporting fatty acids and cholesterols from dietary fat to the body tissue. Then we have 3 other types. So we've got VLDL, which is very low density lipoprotein. That's usually in the liver. It's less tags, and the reason why is because some have already been transported via the chylomicrons, and then they go back to the liver, and then they become VLDLs. And then after that, we've got LDLs, which are your bad cholesterol, and we've also got HDL, which is your good cholesterol. And the reason HDL is good is because it's reverse cholesterol transport. So it's picking up cholesterol and the bad cholesterol and returning it to the liver so that you can excrete it, and you don't want cholesterol. So essentially that's why it's good. The next part we focus on is lipid breakdown and mobilization. So what happens is adipose tissue is where tag or triglycerides are stored. So it's broken down there. And the hormones such as adrenaline and glucagon, so they signal the conversion of fat into monoglycerides and fatty acids. They react with the receptors, and then you break that down, you also get the conversion of glycerol. Oh, what happens is, you produce fatty acids in glycerol, and then the glycerol enters the liver. It's converted into glucose 3-phosphate, which is then used in gluconeogenesis or glycolysis. Fatty acids are essentially, they're insoluble in water. So what they have to do is bind to this serum albumin. And when they bind to the serum albumin, it allows them to be transported in hydrophilic blood. So that's the purpose of the 4th dot point there. And then what happens is, once you get to a tissue, the fatty acid can dissociate from the albumin and enter the cytosol, where it can attach to binding proteins and enzymes. Sorry, I'm rushing. We've only got a few minutes left.
Few minutes.
And the next thing is beta oxidation. So this is how we actually use the fatty acids for energy. So before, we just looked at how fatty acid is broken down from the tissue and it gets to another tissue to be used. So then when it gets to the tissue to be used, it's used via this process called beta oxidation, essentially where it's metabolized. So what happens is, I've got this diagram here. So C16 is basically palmitic acid, I think. It's the smallest one we focus on. So 2 carbons are cleaved at a time, and each cleave produces 1 NADH and 1 FADH2, which can directly enter the electron transport chain to produce energy. What we also have is essentially C16 to C14 is producing a 2-carbon molecule. That 2-carbon molecule is acetyl-CoA. And the acetyl-CoA can undergo, no, it doesn't have to undergo its transition reaction here, which pyruvate does. It can just directly enter the citric acid cycle, which then again produces further NADH and FADH2, which can then enter the ETC to produce more energy. And I think that was all the slides I had here. Next week, we'll be going over the exogenous, endogenous pathways in reference to lipoproteins.
We'll go through in more detail. Like that flow chart, like a few slides back, we'll go through in more detail. Yeah. And there's also one for endogenous pathway, which we'll go through. And we'll go through alcohol metabolism. Okay.
Yeah, so basically there is a lot of biochem coming up, and you'll learn each of the pathways sort of separately, one at a time, and eventually it will be not so scary anymore. So don't be too intimidated by it. It'll be okay. So yeah, both of the recordings, both of the sessions will be up on our YouTube. If you want to spread the word to your friends as well, if they want to get some knowledge going.
Yeah.
Thanks for coming, guys.
Yeah, thanks for coming.